Hello everyone. We are very happy to welcome you in the Educational Symposium this year. This Educational Symposium will be dedicated to the windows of opportunities for nutrition and bone development and will be supported by Nestlé. I will be your chair, and the co-chairs will be Nicolas Bonnet and Marie-Noëlle Orcajada in Switzerland. We are very happy also to welcome outstanding speakers. The first one will be Tatsuya Kobayashi from Massachusetts General Hospital in Boston, who will talk about bone development and its regulation. The second talk will be given by Carrie Cowardin from the University of Virginia about the gut microbiome mediates maternal and nutritional influences on skeletal development. After the two talks, we will have a polling questions. So please wait for the questions, vote. For them, you will have 20 seconds to vote for voting to the questions and then the answer for each of them. After this session, we will also have a discussion and question and answer session. Please feel free to ask the questions through the chat. Thank you very much and enjoy the session. So the first talk will be given by Tatsuya. So Tatsuya, it's your turn.
Good afternoon. In this talk, I would like to review the process of the skeletal development and its regulation, and then cellular metabolism in bone and cartilage. So mammalian skeletal development starts at mid-gestation. For example, in mouse limbs, around embryonic day 9, mesenchymal progenitors in the limb buds start condense and differentiate into cells that later form bone and cartilage. So this process needs to be tightly coordinated to achieve the proper bone development. So mineralized bone form either through membranous bone formation or through endochondral bone formation. Osteoblasts differentiate directly from mesenchymal progenitor cells in the membranous bone formation, whereas in endochondral bone formation, mesenchymal progenitors first differentiate to chondrocytes to form cartilage templates that's later replaced by mineralized bone. So this cartilage template, called growth plate, continuously present in the postnatal bones and drives longitudinal bone growth. In endochondral bone formation, mesenchymal progenitors first differentiate to chondrocytes and as well as in perichondrial cells. Chondrocytes inside further differentiate towards hypertrophic chondrocytes, which induce osteoblast differentiation in perichondrial cells, and they attract blood vessels. Then blood vessels invade into the bone, bringing in osteoprogenitors, and then now bones formed both from outside and also inside. In the growth plate, there are multiple types of chondrocytes aligned in an orderly manner. In the results of population of chondrocytes slowly proliferate and differentiate to vigorously proliferating, columnar proliferating chondrocytes, and then further differentiate to postmitotic hypertrophic chondrocytes, they get mineralized and finally replaced by mineralized bone. So differentiation and proliferation of growth plate chondrocytes are regulated by locally expressed genes. Here I would like to focus on three major signaling pathways, among others, that are particularly important for endochondral bone development. First is BMP signaling, and second is Indian hedgehog PTHRP signaling pathway. And third is FGF signaling and CNP. So BMP signaling through SMAS is essential for the initial chondrocyte differentiation of mesenchymal progenitors. And this is best demonstrated by the genetic study done by Karen Lyons' group 15 years ago, where they knocked out BMP receptor 1A and 1B simultaneously in chondral progenitors. As you can see, a double conditional knockout almost completely lack endochondral bone elements. And mesenchymal progenitors in the double conditional knockouts fail to differentiate to chondrocytes marked with SOX9 and SOX5-6. Instead, they simply just die. So once mesenchymal progenitor cells differentiate to growth plate chondrocytes, different signaling systems control their further differentiation. Particularly, Indian hedgehog PTHRP signaling pathways plays a critical role. PTHRP expressed in reserve zone chondrocytes prevents premature hypertrophic differentiation. And PTHRP expression is dependent on Indian hedgehog's action in the growth plate. In addition to regulating the PTHRP, Indian hedgehog does two other important things. One is that Indian directly acts on reserve cell chondrocytes to stimulate their proliferation and their differentiation. And second is Indian hedgehog most robustly expressed the hypertrophic chondrocytes directly acts on the perichondrial cells to induce osteoblast differentiation in endochondral bones. Thus, the Indian hedgehog couples the growth of growth plates and the bone formation in endochondral bone development. The intracellular signaling downstream of PTHRP has been mapped by multiple groups' efforts in the last 15 years, and it appears that the PTHRP major function is to suppress salt-inducible kinases, particularly SIK3 in growth plate chondrocytes. And 6, then phosphorylate class 2 HDACs, particularly HDAC4 in chondrocytes, to prevent their nuclear localization to block its action. Class 2 HDACs are transcriptional repressors, and then it inhibits the action of MEF2 transcription factors that we know now drives the chondrocyte hypertrophy. Therefore, PTHRP actually inhibits MEF2's action through a cascade of negative regulations. So the importance of FGF signaling pathway in growth plate chondrocytes was demonstrated by the genetic analysis of achondroplasia patients who has activation mutation of FGF receptor 3. And back then, the multiple groups generated multiple mouse models and showed aberrant activation of FGF receptor 3 reduces proliferation of growth plate chondrocytes, mainly through RAS-RAF-MAP kinase pathway. So scientific breakthrough for achondroplasia treatment came from a group in Japan where they figured out how CNP pathway works in the growth plate. So CNP is a small circularized peptide, and as you can see, a CNP knockouts and show a dwarfism like achondroplasia, whereas overexpression of CNP causes a skeletal overgrowth. CNP binds to the receptor NPL2 and drives cyclic ANP-dependent protein kinase, which antagonizes RAS-RAF-MAP kinase pathway at the level of RAF. And this pathway is a major downstream pathway mediating FGF's action. So apparently through this antagonism, CNP systemic overexpression can rescue the skeletal phenotype of achondroplasia. And this discovery eventually led to development of drug, CNP analog, which had been shown to have a remarkable therapeutic effects in achondroplasia patients in clinical trials. So now I move on to osteoblasts. So osteoblasts come from osteochondral progenitors through multiple differentiation steps. So as I mentioned earlier, Indian hedgehog produced by the hypertrophic chondrocytes is essential for osteoblast differentiation in endochondral bones, but not in the membranous bones. Whereas Wnt signaling is universally important for osteoblast differentiations in both endochondral and membranous bones. And so upon receiving the Wnt signaling, osteoblasts differentiate towards hypertrophic, the fully mature osteoblasts that express large amounts of extracellular matrix, such as type 1 collagen, that are major function of osteoblasts. So there are more than 400 genes known to cause skeletal diseases, which are roughly categorized into 42 groups. The defects of several genes are known to primarily affect osteoblasts. These are in these categories. So as you can see, osteogenesis imperfectas are mainly caused by the mutation of matrix genes. But other bone disease are caused by osteoblast dysfunction, such as high bone mass disease, and also low bone mass disease type of osteogenesis imperfecta are caused by mutation signaling molecules, and particularly Wnt-related signaling molecules. So the essential role of Wnt signaling in bone was first revealed by the genetic analysis of patients with osteoporosis pseudoglioma syndrome, where Matt Warman's group identified LRP5, which is a Wnt co-receptor, was causal for this disease. So as you can see, at the age of 10, this patient had a severe osteoporosis and compression fracture. And as I mentioned earlier, the Wnt1 also causes osteogenesis imperfecta type 15, and this girl has all the vertebrates the fracture due to osteoporosis. So Wnt binds to LRP5 and freezol and mainly signals through the beta-catenin signaling pathway. The importance of beta-catenin signaling was confirmed in the mouse models by multiple groups again. And then the loss of beta-catenin in osteoprogenitors leads to a lack of mineralized bone. So in the last few minutes, I would like to discuss about the role of nutrient metabolism in osteoblasts and chondrocytes. And particularly today, I like to focus on energy metabolism and carbon metabolism. So all three major nutrients groups, carbohydrates, lipids, and proteins, are used to generate energy and used to synthesize biomass. The glucose is primary energy source for many types of cells, including chondrocytes and osteoblasts. Also, fatty acids can be metabolized into acetyl-CoA in the TCA cycle, and then the glutamine can be metabolized in the alpha-ketoglutarate in the TCA cycle, and this drives the TCA cycle. The glucose is first transported from the extracellular space into the intracellular space through a glucose transporters such as GLUT1, then metabolized into the pyruvate. And pyruvate can be metabolized into lactate, which generates NAD to drive glycolysis, or pyruvate enters in the mitochondria. So this process of glycolysis generates a small amount of ATP, whereas once the pyruvate enters into this TCA cycle, then it eventually leads to a much more efficient generation of ATPs. So the TCA cycle generates these electron donors to the electron transport chain, which drives the ATP synthase to generate ATP consuming the oxygen. So this process is called oxidative phosphorylation, OXPHOS. In rapidly proliferating cells, such as cancer, prefer glycolysis to generate ATP because glycolysis also stimulates biomass synthesis through pentose phosphate pathways. So the importance of glucose metabolism in bone development has been shown by multiple groups. The Fancy-Longs group has shown that the deletion of glucose transporter GLUT1 the severely disrupts endochondral bone growth with reduced proliferation of proliferating chondrocytes. Guild-Carmeliet's group in Leuven has analyzed energy metabolism in chondrocytes and found that majority of ATP is generated through glycolysis, followed by glucose oxidation in mitochondria and fatty acid oxidation. And glutamine metabolism also contribute a small portion. So glucose metabolism through the glycolysis is the major mechanism for the ATP production in chondrocytes. Cellular metabolism is regulated to meet cell's demand. Again, the Fancy-Longs group showed that osteoblasts prefer to use glycolysis over oxidative phosphorylation for energy production upon osteoblast differentiation. So as you can see, oxygen consumption drops upon osteoblast differentiation from here to there, whereas lactate production, which is the significantly increase upon osteoblast differentiation. This suggests that the mature osteoblast uses glycolysis over mitochondrial respiration. So in fact, they estimated about 80% of ATPs in mature osteoblasts is generated through glycolysis, whereas significant portion of ATP is generated in the mitochondria in pre-osteoblasts. Although glutamine contributes to a small portion of ATP production, nonetheless, the glutamine metabolism is important for bone. So again, the Guild-Carmeliet's group generated glutaminase knockout. So glutaminase is an enzyme required for the glutamine metabolism to alpha-ketoglutarate in the TCA cycle. So as you can see, glutaminase knockout mouse show a reduced bone mass, reduced bone formation, and reduced bone osteoblast markers. So lastly, I would like to discuss about the potential regulatory role of metabolism on cellular function. And particularly, TCA cycle metabolites appears to be important. For example, citrate generated in the TCA cycle can be exported into the cytoplasm and converted back to acetyl-CoA by this enzyme, ATP-dependent citrate lyase, ACLY. Then acetyl-CoA is, of course, used for de novo lipid synthesis, both fatty acid sterols and also the important intermediate metabolites. So these the fat lipid groups are used to modify many types of proteins, particularly involved in the signaling pathways, and to place very important roles. For example, Indian hedgehog is palmitoylated at the N-terminal end and tethered with the cholesterol at the C-terminal ends. And these lipid modifications are essential for its function. So in addition to lipid synthesis, acetyl-CoA serves as a universal donor of acetyl group for many acetylated proteins, including histones. The histone acetylations plays the critical role in regulating the gene expression. So another TCA cycle metabolite is important for has a possible regulatory roles is alpha-ketoglutarate, because alpha-ketoglutarate is essential for many enzymes, including those that mediate demethylation of histone DNA and histones. So together, TCA cycle metabolites might actually directly regulate epigenetic status by providing essential molecules and for the enzymes to work. So for example, in this paper, this group looked at the effects of glucose depletion in histone methylation, acetylation. So glucose depletion quickly downregulates the ACLY and downregulates histone acetylation. So we also generated ACLY conditional knockouts to reduce the acetyl-CoA level. And as you can see, the ACLY conditional knockouts cause lethal skeletal dysplasia with reduced histone acetylation in primary chondrocytes. So I would like to summarize the presentation. So multiple signaling pathways regulate cellular function to coordinate proper skeletal development. And about the metabolism part, cellular metabolism is of course essential, but it may actually have a regulatory roles in osteoblasts and chondrocytes. So with that, I end my presentation, and thank you for your attention.
Hello, my name is Carrie Kawarden from the University of Virginia. And today I'm going to tell you about how the gut microbiome mediates maternal and nutritional influences on skeletal development. And I have no conflicts of interest to report. Evidence over the last 15 years has demonstrated that the gut microbiome can regulate host physiology in numerous ways. This includes shaping immune development and function, neurocognitive development, linear and ponderal growth, as well as metabolism. And multiple factors interact to influence the composition of the gut microbiome. These include diet, exercise, medications, including antibiotics, environment, genetics, and geography. Preclinical mouse models have shown that microbial communities can influence skeletal balance by shaping both bone resorption as well as bone formation. For example, germ-free mice have altered bone volume depending on the timing of colonization, as well as the microbial communities that are used. And antibiotics in early life can disrupt skeletal growth as well. The evidence for gut-bone communication includes the observation that the microbiota is required for bone loss in sex steroid-deficient models of osteoporosis. In these models, supplementation with probiotic strains, including Lactobacillus rhamnosus and Lactobacillus paracasei, can improve bone volume. Microbial communities can also impact linear growth, and supplementation with Lactobacillus plantarum can boost both bone growth via insulin-like growth factor 1 signaling. Human donor microbiota colonization in recipient notobiotic mice can also shape bone volume in those animals. And this is dependent on donor age, with older donors transmitting lower bone volume relative to younger donors. There are three main mechanisms of communication between the gut and the skeletal system, and these include immune signals, microbial metabolites produced by the microbiota, as well as host metabolism, which can be shaped by nutrient processing by the microbiota. All three of these factors interact to influence bone balance, including osteoblastic bone production and osteoclastic bone resorption. One of the most well-known examples of microbial metabolites that can influence bone biology includes short-chain fatty acids. Microbes process dietary components, in this case, a high-fiber diet, leading to production of short-chain fatty acids. These short-chain fatty acids can suppress bone resorption and increase bone production. They can act directly on osteoclast progenitors to prevent differentiation into mature osteoclasts. They can also boost T regulatory cells, which themselves regulate osteoclast formation, as well as enhance osteoblastic bone production. There are also indirect mechanisms by which microbial metabolites can influence bone biology. These include influencing nutrient processing by microbes that influences available nutrition for the host, as well as production of certain vitamins. A healthy microbiota can also protect the intestinal epithelial barrier, leading to reduced inflammatory signaling. And this inflammation can increase osteoclastic differentiation and bone resorption. The microbiota also plays a very critical role in regulating the immune system. And one of the classical examples of this is the balance of T regulatory cell subsets. At homeostasis, the microbiota can induce T regulatory cells that keep inflammatory Th17 cells in check. Th17 cells are known to play a role in increasing the number of osteoclasts and increasing bone resorption. As I mentioned before, T regulatory cells can suppress osteoclast differentiation by suppressing these Th17 cells, and they can also signal directly to osteoblasts to increase bone production. In situations of barrier disruption or inflammation, these Th17 cells become activated, enhancing osteoclastic bone resorption. The microbiota also plays a role in protecting against inflammation by providing colonization resistance. So this is the concept whereby by competing for different nutrients, a healthy microbiota can prevent colonization with pathogens, and those pathogens cause inflammation. Finally, the microbiome can also act as an endocrine organ. Microbial communities are known to regulate host metabolism in undernutrition as well as in obesity. And the context or the composition of the gut microbiota can influence adipose tissue composition independent of caloric intake. As I mentioned before, the microbiota can control IGF-1 production to regulate skeletal growth, as well as produce various vitamins that can be used by the host. The microbiota also plays a critical role in deconjugating bile acids that are produced by the liver. And this is one mechanism by which the microbiota can regulate lipid and glucose metabolism. Clearly, immunity and metabolism are critical to healthy growth, and these pathways can be disrupted by alterations in the microbiota. This is very commonly seen in the context of undernutrition, which is a major global health challenge that affects or that contributes to 3 million deaths annually in children under the age of 5. Stunting or failure to reach linear growth potential affects 149 million children worldwide. Stunting is represented by a height for age Z-score of less than 2 standard deviations below the mean, and it's particularly associated with lifelong negative consequences, including reduced cognitive capacity. Unfortunately, current therapies are really insufficient to produce a robust recovery from stunting. And this is likely because it's driven by many different environmental interactions. So these include not just an insufficient diet, but also local intestinal inflammation that impairs nutrient absorption. And this is commonly because of pathogen carriage within the gut. And those pathogens can exacerbate this inflammation. An immature or undernourished microbiota is causally related to impairments in growth. And this has been shown using notobiotic mouse models of undernutrition. So the microbiota of an undernourished child is chronologically immature for their age, so it looks more similar to the microbiota of a child who's younger. Human milk oligosaccharides are a component of healthy human breast milk, and these are very critical, and breastfeeding is critical during undernutrition. The neutral and acidic milk oligosaccharides make up many of the HMOs within breast milk. And these structures are not digested in the infant gut, and they're actually minimally absorbed by the host. They do have immunomodulatory mechanisms, so they can signal both directly to immune cells as well as indirectly by shaping the composition of the microbiota. 2-fucosylactose is one of the most abundant HMOs in mothers with a functional FU2 gene, which is also called the secretor phenotype. 2-fucosylactose is currently being added to infant formula as a prebiotic and an immunomodulatory agent. Sialylated HMOs are decreased in the breast milk of mothers of stunted children compared to mothers of healthy children. And you can see that here, showing the breast milk of mothers of healthy children in black and mothers of stunted children in white. And there's a significant increase in sialylated oligosaccharides as measured by mass spectrometry in the mothers of healthy children. So we wanted to test the role of these HMOs in host development using notobiotic animal models. To do this, we isolated the microbiota from a 6-month-old stunted, underweight Malawian infant. We cultured many different strains from this microbial community, and arrayed those strains in 96 well plates. We then reassembled the community using isolated strains into a 25-member community that was representative of the original donor. We used this community to colonize recipient germ-free mice and fed these mice a prototypic Malawian diet composed of food ingredients that are commonly encountered in the first years of life. We then supplemented these diets with different milk oligosaccharides to examine the effect that they would have on the host. So we had two different experiments. In each experiment, the mice were originally germ-free. They were switched on to the Malawian diet and then colonized 3 days later with the Malawian microbial communities, and then followed out for another 39 days. In the first experiment, we used sialylated bovine milk oligosaccharides to supplement the diet. So this is a source of those sialylated oligosaccharides. Although it's isolated from bovine milk, they're structurally similar to human milk oligosaccharides. We also used 2-fucosylactose as a model for the current supplementation of infant formula. And what we saw was that both HMO structures were able to significantly influence the composition of the gut microbial community. So SBMO significantly increased the relative abundance of Collinsella aerofaciens, whereas 2-fucosylactose significantly increased Bifidobacterium, Collinsella, and significantly decreased Klebsiella and Peptinophilus. So we do think that these structures are able to influence gut microbial communities and that they have different effects depending on which milk oligosaccharide was used. However, we wanted to know whether these HMOs are able to impact host growth. So looking at weight gain over the course of the experiment, we saw that SBMO actually resulted in significantly increased weight gain relative to the control group. In contrast, we did not see the same effect with 2-fucosylactose. This is again emphasizing that these different HMOs have different effects. However, we wanted to ask whether SBMO is able to impact bone growth because we're really interested in understanding how linear growth is occurring in this model of undernutrition. So we performed microcomputed tomography on the femoral bone from these animals. And we saw a significant increase in the trabecular bone volume in SBMO supplemented animals, as well as a significant increase in trabecular number and a trend towards an increase in cortical thickness. Comparing this to 2-fucosylactose, we did not see any significant difference in trabecular bone volume or cortical thickness after 2FL supplementation. So again, further reinforcing the idea that these different oligosaccharides have different effects, not just on the microbial communities, but also on the host. However, we wanted to ask how SBMO is able to increase bone volume. So we turned to measure common mediator or common markers of bone resorption and bone production. And what we saw was that CTX1, which is a marker of bone resorption, was significantly reduced in the SBMO supplemented animals, whereas P1NP, which is a marker of bone production, was not significantly altered. So this was our first clue that SBMO was primarily impacting bone resorption rather than bone formation. We checked this again using dynamic histomorphometry, in which the mice were administered 2 different dyes that incorporate into growing bone 1 week apart. So we can measure the different distance between these 2 dyes to tell us how much bone was produced in this time period. Again, we did not see any significant difference in bone production between SBMO supplemented animals and the controls by this assay. We also measured insulin-like growth factor 1 and did not see significant differences in IGF-1. So again, suggesting that SBMO supplementation isn't influencing bone growth. We next wanted to ask whether the reduction in serum CTX1, which suggested decreased bone resorption, was stemming from altered osteoclast numbers. We performed tartrate-resistant alkaline phosphatase staining on the tibial bone, which shows osteoclasts in pink. What we saw was a significant reduction in the number of osteoclasts overall, as well as the number of osteoclasts per bone surface in the SBMO supplemented animals, suggesting that SBMO may be reducing bone resorption by reducing the number of osteoclasts. So we wanted to see how this was happening. So we turned towards systemic mediators of osteoclast formation. Osteoclast progenitors recognize RINC ligand on osteoblasts and stromal cells which triggers differentiation and fusion into mature osteoclasts. Osteoprotegerin blocks the activity of blocks this rank ligand signal to serve as a negative regulator of osteoclast formation. Measuring these two mediators in the serum showed that serum rank ligand was significantly reduced in the SBMO supplemented animals, while osteoprotegerin was significantly increased. These pathways directly regulate osteoclast formation, suggesting potential mechanisms by which SBMO boosts bone volume. However, we were really interested in understanding how SBMO is able to induce these changes in systemic mediators of osteoclast formation. So we began to examine the colonic immune environment, seeing if some signals within the gut could originate and then signal to bone cells within the skeletal system to influence bone production or resorption. And what we saw was that cecal IL-1 alpha, so this is tissue levels of IL-1 alpha, which is a pro-inflammatory alarmin, were significantly reduced in the SBMO supplemented group. We also saw a trend towards an increase in IL-10, which is a regulatory cytokine. So this is suggesting that the immune environment in the colon is altered with SBMO supplementation. When we looked by flow cytometry at the immune cells that are making up the colonic population, we saw a significant increase in colonic eosinophils. These are innate cells that are a hallmark of type 2 immune responses. In keeping with this, we also saw a significant increase in cecal eotaxin, which is an eosinophil recruiting chemokine. So this data suggests to us that SBMO is boosting type 2 immune cells within the gut. So at this point, we think that SBMO is acting on this undernourished community and diet context, leading to increased eosinophils and eotaxin 1, as well as blocking osteoclast formation by reducing rank ligand and boosting bone volume by also via osteoprotegerin, which is blocking this rank ligand signaling. However, we weren't clear on what signal was actually inducing these type 2 signals within the gut. So we turned toward RNA sequencing to understand gene expression within the ileal tissue. And what we saw was an interesting gene expression signature that was upregulated in mice supplemented with SBMO. And these include the genes DCLK1, FR3, and the succinate receptor. So these genes were really interesting to us because they're actually a hallmark of tough cells. And these cells are really interesting cells within the gut, and they sense signals in the gut lumen to communicate with nearby cells. And they were interesting to us because they're known to orchestrate type 2 immune responses downstream of microbial metabolites in the intestine. So we next wanted to measure these small intestinal tough cells and see whether they were indeed increased with SBMO supplementation. So we performed immunohistochemistry staining for DCLK1 and identified a significant increase in tough cells within the duodenum, jejunum, and ileum of the small intestine. One of the major signals that's known to increase tough cell numbers is succinate. So we measured succinate by mass spectrometry and noted that SBMO supplementation increased levels of succinate within the cecal content. However, this increase was not present in germ-free animals, only when the animals were colonized with the Mammalian microbiota, suggesting that these metabolites are indeed produced by the microbiota and that they may be responsible for this increase in tough cells. So to summarize, we believe that SBMO is acting on the undernourished community and diet, and this microbiota is producing succinate. The succinate is sensed by tough cells, leading to their proliferation. At this point, we're really interested in understanding how these tough cells are then recruiting eosinophils, or what the signals are downstream of these cells. We're also quite interested in understanding whether there's a connection between these eosinophils and type 2 immune responses with the systemic mediators of bone formation bone resorption that we've observed being decreased in the SBMO supplemented animals. So these data are quite interesting and for several reasons. So the first is that maternal HMOs are causally linked to growth in this model of undernutrition. So the composition of breast milk does seem to matter in terms of growth. We also see that different HMOs have specific effects on host biology. And this is critical because it allows us to identify specific roles for different HMOs to improve the design of infant formula. formula. So one size may not fit all. We may need to tailor our approaches depending on the context. We also see that the microbiota is required for the full effects of HMOs on host biology. And this could allow us to isolate strains that utilize SBMO, which could eventually lead to more useful probiotics. So with that, I want to thank the Gordon Lab at Washington University in St. Louis, where this work was performed. I also want to thank some of my current colleagues. I'm now an assistant professor of pediatrics at the University of Virginia. So I want to thank everyone else who's a member of this microbiome initiative, as well as my own lab members. And with that, I'd be happy to take any questions.
Thank you. So we'd like to thank both speakers about their very nice talk, and I'm sure that there will be a lot of different questions about those talks. So now, if you don't mind, we will go through the polling question, if we could have them. Okay. So remember that you can use the Slido to vote. You will have 20 seconds to do that, and then we'll give you the answer. So the first one is, what nutrients is mainly used for energy production in osteoblasts and chondrocytes? So seems that everybody followed. Just remain few seconds. Okay, and the answer is the answer B, glucose by glycolysis and fermentation. So most of them answer well. So we now switch to the second question. Okay, so the second question you can read and don't hesitate to vote, is what signaling pathway is negatively regulated by CNP and PR2 signaling in growth plate chondrocytes? So it seems that, okay, so a few seconds. Okay, 2 seconds more. Then, so the right answer was the FGF signaling. And then maybe Tetsuya will provide the explanation of that again. So next, we'll go to the third question now. So the third question is, what are the major pathways by which the gut microbiota influence bone balance? And so you can start voting. You have different answers: immune regulation, microbial metabolite, microbial colonization of the bone marrow, or hormone production and signaling. You will be able to vote for several questions. One or several questions are right. Okay. So I think we will allow 3 seconds more. Okay. So the answer is the microbial metabolites, immune regulation, and hormone production. So this is good. And the last questions will be, so how might human milk oligosaccharides increase bone volume? So you can start voting, and you can vote for one answer or several ones. And you see the different answers. Okay, we will allow a few seconds more. Seems to be very clear. for most of you? Okay. Okay. So the answer is the answer D, have different effects. So the talks were probably extremely clear, and congratulations to all of you that have answered very well. So now it's time for question and answer, and I will give the floor to Nicolas and Marie-Noël, and I'm sure there will be several questions for our speakers.
Yeah, so congratulations to two speakers for this really nice presentation. And actually, I have the first question for Professor Kobayashi. There is one mention that there is a recent study which point out previously the role of marrow lipids in regulating bone remodeling. Could you speculate on the role of adipocyte during this key event of bone development?
I don't know much actually about that. So clearly they are coming from the same sort of mesenchymal progenitors. When signaling the works in the opposite way to the osteoblasts and the adipocyte differentiation. But that's, I'm sorry, I cannot tell you much about how adipocytes actually influence the osteoblasts in the microenvironment. But it's very well known that they usually move reciprocal way when the adipocytes go up and osteoblasts go down, except a few exceptions. Where based on some genetic strain, some mice have both high bone mass and high adipocytes in the marrow. So I don't have a knowledge to answer your question and how they were interacting each other. I'm sorry.
Okay, thanks.
So thanks a lot to both speakers for the very nice talks indeed. So one question for Carrie is that actually, is there data on the metabolic pathway that are active in microbiome and immune cells that may explain the nutrient competition?
Yeah, that's an interesting question. We don't know specifically about the metabolism in this model, but I think it's a good point, because that's certainly going to be strongly affected by diet and strongly affected by how the microbiome is processing the diet as well. So that would be something that would be really interesting to look into.
Okay, so second question for Professor Kobayashi. Vascularization is a key event in bone development, and actually we would like to know if Indian hedgehog and or PTHRP can influence the appearance of the primary and or the secondary center of ossification.
So Indian hedgehog PTHRP signaling regulates chondrocyte differentiation, but not directly influence the vascularization. So one of the major factors that attract blood vessel is a VEGF produced in hypertrophic chondrocytes. So in that regard, PTHRP regulation indirectly regulates the VEGF expression by regulating hypertrophic chondrocytes, but it has no direct regulation.
Okay, thanks.
So a question for Carrie. Do sex hormone influence microbiota bond communication? And if yes, how does it occur?
I would say yes. So I think there's certainly been shown to be a role for the microbiota in sex steroid deficient bone loss. And different microbial species are going to have different effects, and so supplementation with various probiotics has been shown to have an effect on bone volume in those models. I think the point about potentially sex steroid differences driving microbial composition is a little bit less well worked out, but would be certainly something that would be worth investigating.
Okay.
So the third question for Professor Kobayashi: are chondrocyte characteristics altered in your COL2 mutant mice?
I'm sorry, say that again.
Are the chondrocyte characteristics altered in your COL2 mutant mice?
COL2 mutants. You're talking about the mutations according to gene?
Yeah.
Well, so they call, of course, it's according to mutation causes the chondrodysplasia, the achondrogenesis. Yes. I'm not sure it directly changes chondrocytes, but it changes the cartilage more, the function due to the dysfunction of matrix generation. I'm not sure I'm answering your question.
No, that's okay. Thanks.
Okay, so a third question to Carrie. So as you can see on the screen, actually, there was a question around the effect of HMO, if it was detectable on not undernourished kids, and if not, how strong should be the undernutrition be for HMOs to make a difference?
Yeah, I think that's a great question, and that's something that I would love to test. We haven't tried the specific HMOs in different models of development, so a not undernourished model. But I think that would be really interesting to test. We do see that SBMO in germ-free mice is able to increase bone volume slightly, but not to the same extent as it does in the presence of the microbiota. So I think all of the evidence is kind of pointing to the idea that the microbial context and diet context are probably both important. So I think that would certainly be worth looking into. And in terms of how strong should the undernutrition be for HMOs to make a difference, I think that kind of goes along with the idea that we might have different effects based on the extent of undernutrition. In this case, we're using a model where the donor microbiota is from a severely stunted and underweight infant, and so it's a pretty robust model of undernutrition, and it would be interesting to kind of see if we could maybe come up with a system that's a little bit more mild, or just mimicking other conditions that these kids might be experiencing.
Thank you.
Okay, so for Professor Kobayashi, in terms of nutrition and energy deficiency, which are associated with delay of growth, would you recommend to use glucose, fatty acid, and or protein to boost the cell energy?
So whether the nutrition can be a therapeutic target. It's a interesting question, but for example, during the embryonic development, the diabetic pregnancy causes overgrowth of fetus, but when you look at the overgrowth of the fetus, it's mainly the adipose tissue and muscle are the ones that influence, but not the skeleton or brain, because it's caused mainly through the insulin signaling, but not the glucose per se. So this evidence that other pieces of evidence suggest that it's probably difficult to directly influence the bone growth through changing the nutrition.
Okay, thanks.
So, Professor Corwin, at the beginning of your talk, you also mentioned briefly that the microbiome and the bacteria are involved in the production of vitamins, can be involved in this. And do you have any insight on how this translates, which vitamins and how this would translate into bone benefit?
Yeah, I think that's a really interesting question. I know that the microbiota, can produce vitamin K and I think vitamin E as well. I think there are some links with vitamin D metabolism, but they're maybe not as well worked out. So in this particular system, we haven't looked at vitamin levels, and I do think it could be particularly interesting if there was an effect on like absorption of vitamins as well. So I would say I have my knowledge of those pathways is a little bit limited, but hopefully this has given you a couple of ideas about things to think about.
Thank you.
So another question for Professor Kobayashi. Actually, can you precise the type of glucose transporter which are really expressed in chondrocyte versus osteoblast? And are there more insulin dependent or independent?
So the major glucose transporter expressed in the chondrocytes and osteoblasts is GLUT1.
Okay.
And then GLUT1 is not insulin sensitive. So glucose transport of osteoblasts and chondrocytes is probably not regulated by insulin signaling. But the recent studies show that the expression level of glucose transporter can actually stimulate glycolysis. So the expression level of these glucose transporters is probably one of the major determinants of glucose flux.
Okay, thanks for this nice precision.
So there are some other questions related to what you reported with the SBMO. And one simple question was, did you ever test the SBMO on bone metabolism in adults or old mice?
Yeah, we have not tested it in aged mice. Our model that we were using, the mice were followed from like 4 to 9 weeks of age. So technically, a lot of the samples were examined at maturity, so at 9 weeks, but we have not tested in older mice. And I do think that would be a potentially very interesting thing to test, particularly maybe in like an osteoporosis model or something like that.
And what would be the mechanism you would think that would explain a benefit on limiting the bone loss if it's in an ovarectomized model?
So I would speculate that potentially could reduce bone resorption. We definitely see effects on kind of immune tone within the gut, and we think that is influencing bone biology. And so if there's inflammatory signals that are increased in osteoporosis models like Th17 cells, perhaps there could be some sort of counter-regulation with the type 2 immune responses that we're seeing that are boosted by SBMO. But of course, that's all speculation. We don't have any data to support that at the moment.
Thank you.
Okay, so another question to Professor Kobayashi. Actually, can you elaborate what would be the effect or the extent effect of a Warburg effect in osteoblast differentiation?
About Warburg effects?
The Warburg effect.
So that's also called aerobic glycolysis.
Yes.
The findings are discovered a long time ago. Mainly the cancer cells uses glycolysis to generate ATP even in the presence of oxygen. So I think I do not know much about the mechanism. One of the mechanisms may be the HIF1. So cancer cells regulate the overexpress HIF1 oftentimes, which prefers which positively regulate glycolysis and negatively regulates mitochondrial respiration. And then the Warburg effects, as I briefly mentioned, have advantages of shunting glucose metabolites into phosphopentose pathway, which generates ribose and NADPH, which are required for lipid synthesis. So the speculation is that the glycolysis is preferred to use in those rapidly proliferating cells in order to use glucose as a biomass, synthesis.
Okay, thanks.
Not really the energy production.
So another question to Professor Kawadin. So what has been, in the effect you reported in your talk, some similar effects on bone, similar effects have been attributed to extracellular vesicles in milk. Could this be a confounding factor of the observed effect on the bone that you showed us?
Yeah, that's interesting. I think, it depends on the preparation. So the 2FL that we used was recombinantly expressed in E. coli, whereas the SBMO were purified from bovine milk, and they are, highly purified, but it's possible that there could be, I don't know the likelihood of there being extracellular vesicles within the purification. It's not something that we've looked at, but I'm open to the idea. So it could be something that we should check. But again, it's going to be highly dependent on how you prep the different oligosaccharides, and so if that's a concern, then it would certainly be worth testing for that, and also planning for how the purification occurs to try to minimize something like that.
Okay, so there is another question for Professor Kobayashi. Since glucose metabolism can impact osteoblast differentiation and function, can tumor cells would modulate osteoblast glucose metabolism in order to affect the colonization?
I have no answer to that. So in my so the Fancinron actually knocked out GLUT1 in osteoblasts, and it has a relatively modest effects in the bone in normal settings, unless osteoblasts are stimulated with Win7 or something. So osteoblasts probably have some other means to generate energy, such as glutaminolysis. So it's not really totally dependent on the glucose for their lives. So the tumor might actually influence the osteoblasts to favor their metastasis in the local environment. But the mechanism through the glucose availability is unlikely, in my opinion.
Okay, thanks.
So another question to Professor Kowadin. So if we consider the HMOs as prebiotics and so on, do you know if there are specific bacteria families that has been shown to better, let's say, degrade these HMOs, and then that could be linked to a higher bone benefit in the future?
Yeah, absolutely. So I think, obviously one of the biggest consumers of milk, ingredients in general would be bifidobacterium, which is very prevalent in early life, and they're known to have utilization pathways for different HMOs. Interestingly, in this model, the primary gene expression or microbial gene expression changes were actually in Bacteroides fragilis and E. coli, and so there was some of the work from a colleague of mine previously showed that there was some cross-feeding interactions between those two bacteria where I believe the Bacteroides was degrading some of the milk products, and then they were actually boosting E. coli levels via that process. And so it's like many things with the microbiome, it's really complicated, and there's a lot of interaction going on, and so it does take a great deal of data and thought to try to tease those interactions apart. But the idea that we could kind of use this system to identify microbes that process different dietary ingredients but then have different effects on the host, I think is really valuable.
Thank you.
So another question for Professor Kobayashi. Does the knockout of aglycine in chondrocyte alter the characteristic of the chondrocyte per se?
We have looked at histology at the moment. I haven't done the thorough characterization. So the growth plate looks relatively normal in terms of structure. But at the cellular level, the growth plate chondrocytes have a less flattening of the characteristic of proliferating chondrocytes. And at cellular level, the chondrocytes might have some inclusions, which we haven't been able to characterize yet. So there are changes in terms of tissue. The structure of the growth is relatively preserved.
Okay, thanks.
So, to Professor Corwelling, coming back a little bit on the short-chain fatty acid, what would you rank as the most documented one in terms of bone benefit and on bone cells impact with the succinate, butyrate, propionate, acetate? What would be your thoughts on this? Which one would represent the best, or is it the combo of them, or one of them that is more active than the other ones? Or?
Yeah, that's a great question. I think butyrate would probably be, would kind of jump out at me as being maybe the most well understood in terms of, immune interactions and potential bone interactions. In this case, we see elevated succinate in our model. We haven't connected the succinate directly with the skeletal development. And so we haven't actually tried supplementing with succinate to see if that boosts bone volume, but it would be really interesting to try that. And I think that's something that needs to be done. And, I think just as we learn more, we appreciate how, specific short chain fatty acids have specific effects as well. So it's just a kind of complicated interaction between many different players in the microbiome.
Thank you.
Yeah, probably a last question for Professor Kobayashi. What kind of role could have the oxidative stress in the context of metabolic switch from the oxidative phosphorylation to glycolysis in the osteoblast maturation process?
I have absolutely no idea about that. I'm sorry.
That's okay.
That's yes, I have no idea.
Actually, do you have any idea about a specific signaling pathway which can, I would say, push this switch of the oxidative phosphorylation to glycolysis, or vice versa, a switch of the glycolysis to oxidative phosphorylation?
Well, so many growth factors stimulate glycolysis for the obvious reasons. Then the switching is, so for example, stem cell differentiation, usually the stem cells are more reliant on oxidative phosphorylation rather than the glycolysis. Then once they start proliferation, then their glucolytic genes be robustly upregulated and from promote glycolysis. And so the many types of growth factors actually stimulate glycolysis, and insulin signaling is of course one of them. But how they are switching, I actually don't know. Someone must know.
Okay, thanks for this nice precision.
And so maybe a last question to Professor Corwadin. Actually, malnutrition is also present in elderly, and how this affects the microbiota composition. Is it different than in babies or same?
That's another really good question. We certainly know that the microbiota changes with age. And so, over the first 3 years of life, infants and children go from, essentially no microbial colonization at birth to a progressively more complex microbial community that looks similar in configuration to an adult microbiota around 3 years of age, maybe a little later. And then, of course, as an individual matures, an aged microbiome could also potentially have an effect on various aspects of biology. And so I think it's a very good question. And I don't know in our specific system whether we would be able to use an aged microbiome to ask whether HMOs might have an effect there, but it's a great idea.
Thank you.
Okay, so thanks. I think that we are arriving at the end of the symposium, and I would like to take just 1 minute really to thank the two speakers, Professor Kobayashi and Professor Corwadin, because actually they are in US and they wake up very early for us in order to be really to have this question and answer really active and really alive. So really thanks a lot for your two talks and this nice discussion, and actually for all the questions that we did not have enough time to ask you. We will try to give you an answer as soon as possible. So I think that with my co-chair, Martin Cohen-Soal and Marie-Noëlle Orcajeda, we really thank you for this nice session. And I hope that you will enjoy the rest of the meeting.
Thank you.
Thank you.